of those, there's a couple slides in there that lists identifying properties of these things. For example, and I might not remember all of them, so you got to go look in the notes for this. Alpha helices are right-handed helices, meaning they turn in a specific direction. Alpha helices have 3.6 amino acid residues per turn of the helix, or something like that. And, what amino acid are you never going to find in an alpha helix? Proline, because it has a nickname of a helix breaker. So there's little facts like that in chapter 6 that you need to know about alpha helix and beta sheet. So focus specifically on the facts about alpha helix and beta sheet. Yes?
So if we were asked like which level of structure like the protein is folded, that's secondary?
That is a little bit vague. They'll make it more clear, like what level protein structure is your protein first functional, and that's the next thing I'll get to. So after you study your alpha helices and your beta sheets, you will learn a couple things about them, is that they are held together by what intermolecular force? Hydrogen bonds, and only hydrogen bonds. When we're talking about secondary structure, we're only talking about hydrogen bonds that hold them together. In fact, there might be one problem-solving type question regarding that. And I went over this at SI today, is that if you are given this sequence and you are told it will form an alpha helix, between which two amino acids, or what amino acid will be hydrogen bonded to aspartic acid in the helix? It turns out that there's a rule regarding alpha helices, and it is called the N and N+4 rule, which is just another way of saying, when you look at, in this example, aspartic acid, make 4 hops, so 1, 2, 3, 4, and the 2 amino acids that will be hydrogen bonded together will be leucine and aspartic acid, okay? The N to N+4 rule is you just make 4 hops down to the right to figure out what amino acid is hydrogen bonded to the first one, okay? Maybe if he was asking instead which one is hydrogen bonded to methionine, you make 4 hops to the right, 1, 2, 3, 4, and it would be alanine in that case. Okay, so that is a rule specifically for alpha helices, is that a hydrogen bridge or hydrogen bond is formed between an amino acid and one that is 4 steps to the right, 4 steps to the C side of that amino acid. Okay. All right. That's alpha helices.
So they're 3.6.
Somewhere in that chapter, and it might be in 6A, I'm not sure, they talk about an experiment called Anfinsen's experiment, right? Anfinsen's experiment, what that did is it proved that the next level of tertiary structure, I'm going to talk about the tertiary structure of a protein, which is your 3-dimensional first functional unit of this protein, is your tertiary structure. The only thing that determines that 3-dimensional structure is what amino acid sequence there was at the primary level. So what Anfinsen's experiment was is that there is this 3-dimensional enzyme, I think ribonuclease. This guy unwound this protein using a bunch of different chemicals all the way down to its primary amino acid sequence. The result of the Anfinsen's experiment was that when he removed all those chemicals, that amino acid sequence by itself wrapped into its original 3-dimensional shape and still had the function that it did initially. So the experiment proved that the functional part of a protein, meaning the 3-dimensional part of a protein, is dependent simply on what amino acid sequence was there at the primary structure level. Okay, so that's what Anfinsen's experiment was. That's what it proved. Okay, so make sure you know that. Okay, good. Question? Yes. So the tertiary structure depends only on the primary structure amino acid sequence. Okay. All right. All right. When we get to our tertiary structure level, we now have different intermolecular forces that keep it together. Other than just hydrogen bonds, we now have a bunch of different things. We have disulfide bonds now at the tertiary structure. Disulfide bonds, remember, are between what type of amino acid will form disulfide bonds? Cysteine is going to form disulfide bonds. You also have, other than hydrogen bonds and disulfide bonds, you have ionic bonds, meaning if there's a amino group on an amino acid somewhere, and there's a carboxyl group on some other amino acid, they will interact via an ionic-like interaction. So ionic bonds will help. There's another type of bond called hydrophobic interactions, right? I think that's in chapter 1 or 2. All hydrophobic interactions are just interactions between nonpolar molecules. So what category of amino acids do you think is most likely to perform hydrophobic interactions? The nonpolar amino acids, okay? So little clues like that, yes?
So for the ionic bond thing, you gave an example of how where you had like an alpha helix and the N and C groups were forming kind of like bridges. Is that the kind of ionic?
With the alpha helix, the N and C groups in the backbone, that's actually the hydrogen bridges that we just did in the previous problem. That's what that one is. It's hydrogen bonds. Yeah, that one's hydrogen bonds.
In SI, you said when you have like two amino acids and they're associated with each other,
Yeah.
that's like an ionic. But you said when they're bonded, it's not. So why not? What's the bonding thing?
So and I'm not really sure in which context you want in this scenario, but if they're not directly connected to each other, it is a weak intermolecular force ionic association. If they are directly bound together like sodium chloride, that is a strong ionic association. Okay. Yes.
Okay, so you said that for the disulfide bonds, that was a cysteine that does that?
Yes, between two cysteines. It's the only amino acid that can form disulfide bonds is between two cysteines. Okay. Yeah, so let me make sure those are the intermolecular forces here. Hydrogen bonds, disulfide bonds, ionic bonds, hydrogen bonds. Those are the one, and I said hydrophobic bonds. Those are the ones that help you determine the three-dimensional shape of an amino acid sequence. That is what causes an amino acid sequence to fold into its three-dimensional shape. So just wanted to clarify that. And note, of that list, one of them was a covalent bond, which on that list there is technically classified as a covalent bond. Disulfide bond is the only one on that list that is a covalent bond. Everything else is either ionic or intermolecular. And in this case, ionic is that weird category where in this class we call it an intermolecular force. Okay, yes? You, yeah.
Oh, you said the rest were ionic or what?
So the only let me simplify it another way. The only covalent bond that holds things together at the three-dimensional level is a disulfide bond. Everything else is classified under the umbrella term non-covalent.
Okay. And then you said that all of those bonds help fold. So do hydrophobic interactions help it fold more?
Yeah. Think of it another way like this. If your amino acid sequence has a mix of polar functional groups and non-polar functional groups, when the protein folds in on itself, it's going to hide all those non-polar functional groups on the inside of the protein, away from the water that this protein is floating around in. So that's what we mean by hydrophobic interactions, is that the hydrophobic non-polar amino acids are going to fold and be hidden on the inside of your protein structure at the three-dimensional level. All your polar stuff, acidic and basic stuff, are going to be on the outside of your protein because that's where all the water is. So they're going to interact with the polar environment of the water. Okay. So that's what we mean by hydrophobic interactions affecting the three-dimensional shape of a protein. All right. Almost done. Last bit of information that's important are, and I think this is in 6b, examples of tertiary structures of proteins, and that is alpha keratin, beta keratin, and collagen. These are your three characteristic examples of structural proteins that are at the tertiary level. There is lots of facts about each one of these. The most important ones for each one of these are listed here. Since we're a little bit over time, I'm going to let you guys go look in your notes. They're not hard to find. Each of these facts is listed somewhere in there. Make sure you know which one is associated with which of these structures, okay? Yes?
Can you quickly explain that?
Yeah, let's see. F is sequence info. All right, yeah, sure. When you go look at beta keratin, there's a specific pattern that sequence has that makes it form beta keratin. For beta-carotene, it is the very simple repeating pattern of glycine, alanine, glycine, alanine, glycine, alanine forever, or glycine, serine, glycine, serine, glycine, serine forever. That's somewhere in your notes there. What that ends up causing is that when you go look at a structure of beta-carotene, all of the glycines are on one side of that amino acid sequence, of that three-dimensional amino acid sequence, and all the alanines are on the other side. What that allows you to do is stack a bunch of these together, and you get a very silky smooth texture. Yeah.
Would anything give you glycine, alanine, glycine, but on like one plane and glycine, alanine, glycine?
No, they'll match to each other. You can look at the picture they have for beta-carotene. That's the only example.
Glycines and alanines all plane on opposite planes?
It's hard for me to picture what you're saying there. Just take a look at the picture they have there, and that's it. So make sure you study those components there, and let me make sure. And yeah, that's it for now. If it's able to be uploaded, it's limited by how long it takes for that. All right. Yep.
Yep, no problem.